Thank you all for coming. I'm really excited to be hosting this event. My name is Chad Collier. I'm the North American Consumables Manager for Bionano Genomics. So today I'm just going to give a brief introduction about what we think Bionano's role is in this genome assembly space, and then we're going to get right on to some of these great talks that our speakers are going to give for us, okay? So, we want to recognize some of the challenges that are in the de novo assembly space, and I think we can recognize there are a few key elements, right? So accuracy is the first thing, right? We want read-lings that are very, very accurate so that we can distinguish these unique and repeated sequences throughout our genome, and then homologous chromosomes as well. Then we also need to have sufficient read lengths, right? So we want to be able to span these repeat arrays and get through it, but ideally we'd have a combination of both of those, right? We would love to have a single contiguous perfect read that goes from end to end on a chromosome. Barring that, it would be great to be able to get, just through some of these larger repeat expansions that are there, requiring about a 3 to 5 megabase ability to do that. We're not quite there yet. So what have we done to sort of get around that? We're using some of these core technologies that you're all familiar with. So Illumina is in the game because it creates very accurate reads, right? They're very famous for that. And those reads are up in the KB range. And then we've got technologies like PacBio, Oxford Nanopore 10X that are delivering longer contigs and scaffolds in the 1 to 10 megabase range. And then we've got sort of a huge leap up to Bionano. Our maps are in the 20 to 200 megabase range. So contiguity is a big feature of our scaffolds, and we're very, very excited to sort of offer those. And then there's folks using Hi-C to sort of span centromeres or regions that other technologies aren't able to do. So, a combination of these technologies has been a feature of a lot of the talks that I've seen this week. And so I want to focus on a few ways that Bionano has been used in some of these projects to really deliver simplicity or increased accuracy using our Bionano whole genome imaging system. And so, sorry I have to lean over a little bit to see this, but this is really a basic example of how our technology is used. So this is a Bionano map here in blue. And then we're just using that very, very long map to scaffold these sequence contigs in the correct order. I love this example here. This is from a GOAT paper a couple years ago. And what we're showing is the Bionano map here in blue. This is the hybrid based on these two contigs plus the Bionano map. And what it looks like is that this particular contig has pretty great contiguity with this section of the hybrid, but we're seeing this contig also shows like it should be there, but there's, what we're recognizing, there's very likely a chimeric join there. And so using the power of the Bionano molecules that are there that were used to build this particular map, we can go back and check, is the problem on the Bionano side, or is this really a chimeric join? And so we've got ample support here with all these molecules from your particular sample that were used to build a map. We can clearly say, okay, yes, this is a chimeric join. Let's break it, and let's take this piece and integrate it elsewhere in the genome where it belongs. So again, we're improving contiguity. We're also improving accuracy. And then this is an example here of a couple technologies being used. Hi-C is in the mix here. And the Bionano map clearly shows that the Hi-C is making a bunch of inversion errors, and then that's been validated through a third sequencing technology here. So the power of all of these sort of improving the overall accuracy of these assemblies that we're going to be delivering to our research community, right? Oh, it's not good? Oh, sorry. Okay. This one. Yeah. All right. Is that better? Yeah. Oh, sorry, guys. Yeah, okay. I'm in the zone. Okay, so. So this is an example of PacBio underestimating insertion size lengths. And so again, this is compared to Bionano, we're sort of hammering home this idea that the complementarity of these technologies is huge and that we should be using them to build better reference genomes. So this is a story from one of our customers at NCI Frederick, and I like it because it really hammers home the idea of the simplicity angle that Bionano is able to provide. And so she uses an analogy of like Bionano, when I building a Bionano map is much like building a 10-piece puzzle, and I'm using these other technologies from like Illumina, very tiny reads. It's like trying to put together a 1,000-piece puzzle. And so that I think that notion of simplicity, combined with the notion of higher accuracy is really, really an important message, and I think we've all sort of seen that it's necessary to do this. And so how do we do this? What specifically is Bionano using to do this? And so we've built the entire stack to deliver from sample to Bionano map. So we've got sample prep and DLS labeling kits, or excuse me, just labeling kits in general, to take your high quality DNA that you use made from our sample prep kits, and then label them specifically in the way we need to run on our Sapphire chip, which is then loaded into the Sapphire instrument. And then we have a suite of software called Access and Solve that is used to analyze and then visualize that software, or those results. So what does our workflow look like? Workflow's pretty straightforward now. We've really had a lot of development work hammering out a lot of the rough edges over the past years. So we're going to start with, if we're going to look at high quality, ultra high molecular weight DNA, we need to isolate ultra high molecular weight DNA, and there aren't a lot of methods out there to do that. So Bionano has been particularly busy developing really great methods for this. And so we've got sort of our standard plug lysis preps, but then recently we've also added this SP prep for blood cells and bone marrow aspirate samples, which delivers those same sort of high quality molecules in 4 hours now. So that's a significant improvement, and we're working to sort of move that into other tissue types over the coming years and months. So once we've isolated the DNA, now we need to label it. And so in the literature, you're going to see mention of both NLRS and DLS now. So we're starting to see DLS pop in the literature. But our previous labeling technology was called NLRS. So that uses a nickase enzyme to nick a single strand of the DNA. And then we do a repair in the presence of labeled fluorophores to add a label at this specific sequence motif, sort of similar to restriction digest, but again, it's not completely cutting the DNA, it's just nicking it. But we had a massive sort of quantum leap forward when we introduced DLS, because instead of damaging the DNA, we're all about trying to maintain contiguity. So we're not using our own tech to sort of damage the DNA now, we're just directly applying a label at a similar sort of sequence, 6-base pair sequence motif that's scattered randomly throughout these genomes. And so now we've got long DNA, we've labeled it, we're going to put it into the Sapphire and the Sapphire chip. The Sapphire chip has been engineered to linearize this DNA and confine it discretely into a nanochannel. And by doing that, we're able to run these labeled DNA, run the labeled DNA in cycles to generate enough DNA or enough sample to accurately represent your particular genome at the read depth that you're after. We take those physical images of the molecules, convert them into a digital representation of the molecule, and use those to use a single assembly algorithm to de novo build a consensus genome map for the particular organism, which is super useful. You can use it for, we've got an example here of, I think it's detecting an insertion, so that's a structural variation pipeline, but then these are also very useful scaffolds for building out your genome assemblies. So this week, there was a lot of talk about N50s this, N50s that. Contiguity is not necessarily the be-all, end-all for genomes, but accuracy is really important. And so I want to talk a little bit about first contiguity improvements, because those are awesome. This is an older slide. We released this during our after DLS first came out, but I think it's really still really great to sort of see a maze genome going from a 1.2 megabase N50 up to 100 megabase N50, but then also including 99.5% of the NGS contigs in that scaffold, which is really fantastic. And so we've got lots and lots of examples that look like this, and we can definitely talk at length about how a particular genome has been improved by integrating a Bionano map. The other day, though, Kelly Daw was specifically talking about using these Bionano maps to build out really high quality genome representations of the 25 founder lines and then 25 NAM founder lines. And the amount of discussion that was in that workshop was really fantastic. It's really exciting. It's very early days for these guys, but the amount of downstream stuff that the community wants to do with this resource already is really exciting. So we're very happy to be a part of that work like that, and continuing to see it happen. So this is just a quick jumble of a lot of the organisms that we've already run, and I was joking, saying that in a few years we're going to need either smaller font or a bigger screen, so we'll see which happens first, right? But we've really been involved in a lot of various assembly projects with you guys, so it's so exciting. Thank you for that. This is a specific one that just came out real recently, so I thought it was one of the just a few weeks ago. So this is a an example of this multi-omics approach, the kitchen sink approach, including optical mapping, that resulted in some really spectacular assembly specs for the Indian Cobra, right? So this is done by the folks up at Genentech, and I was talking to Kushal, the guy who was running this, and he's very excited to be able to say this is a resource that they're going to be able to use in conjunction with, they hope, with other venomous snake projects to maybe develop a broad spectrum antivenom, which is really super useful for human health, and then also there's also some other benefits for like hypertension and pain too. So again, Bionano, a part of that, very, very exciting to see it happen. The second group that's also using that kitchen sink approach, we've got some members in the room, is the VGP. So they've been, we've been a partner with these guys for quite a while, and it's really, really exciting to see us, Bionano as part of this. Really, their goal is amazing, to be able to build error-free reference genomes for the extant 70,000 vertebrate species on the planet. That's a huge mandate, and we're happy that we can make their lives just a little bit easier. And so we've got some resources on our website if you guys are interested in that. But then also there's I think Wednesday, you guys, VGPs has a day-long workshop. So they'd be happy to have you if you want to be a part of that. So this is probably going to go to the next slide. I'm not sure I fixed it yet, but over the past few years, we've had several Nature publications happen, and it's again sort of illustrating this notion that Bionano is a foundational technology for the work that you guys are doing. In this room, I believe, we have some copy of our publication list and then a recently updated version of our hybrid scaffolding white paper. So those are great resources for figuring out any question or answering any questions that you have. Along with, I see tons of my colleagues here too, so they'd be happy to answer any questions. Or if you don't have time, visit us at booth 325. We'd be happy to talk to you there too. Okay, so before I quit, I'm going to just spend 2 seconds talking about some recent updates that we've had on our genome informatics side. So through 2017, 2018, our team worked really, really hard improving the hybrid scaffold algorithms to really be able to deliver better quality for you guys. And so in 2019, they took the same sort of care to improve a lot of the tools that we're using in the human side for IDing structural variants in the genome, and new ways to sort of compare those across large sample sets, and really digging into the amount of power that's available when you're looking at larger sample sets. And so we're really excited to sort of see and deliver that in 2019. In 2020, there's been a commitment to try to deliver some of these tools to the non-human populations, make them work better for non-human samples. So that's, I think, really exciting and something to watch for. So, stay tuned for more announcements there. And then we've also had some recent community efforts too, which I think have kind of flown under the radar a little bit. So I don't have time to really get into any of these specifically. The slides are going to be available afterwards. So, if you see anything here that seems interesting, please come back to the list, dig in, and find out what it is that these guys are doing that maybe something specifically that our software is not able to provide, that these guys found a reason to build a tool to do. Okay? So, just quickly, we're showing the true structure of the genome. We're agnostic to sequencing technology used, no matter what technology you've used, we're going to make it better. And we're the only non-sequencing based scaffolding technology that's capable of correcting sequencing errors, which we think is super exciting. So we're happy to be a part of this work that you guys have been doing, and we hope to continue. Okay, thank you.